Okay, this is going to be the last lecture in this marathon session of lectures today. This one is just going to be nucleic acids. I think what I've decided to do is actually talk about proteins in class, because I want to be sure we got a great understanding there. So, jumping in, and I'm actually immediately going to pause. Hopefully that worked. I'm dumb. Okay, nucleic acids. So, they are DNA and RNA, essentially. Those are the two ones that we have. And they are also polymers. Their monomers are called nucleotides. And I'll talk more about what they are composed of on the next slide. But the main atoms involved here are carbon, hydrogen, and oxygen, the usuals, as well as phosphorus and nitrogen. Nucleic acids are one of the main reasons why we need phosphorus in our diet. The two main types of nucleic acids is DNA, which is the way we store genetic information in order to eventually make proteins. And RNA are they do a lot. RNA is actually really way more interesting than DNA. They're involved in protein synthesis as well as regulating other genes. We're going to talk a lot about RNA later. For now, just think of them as the middle step in central dogma. Remember, you go DNA, RNA, protein. The nucleotides, the monomers of these, have three parts to them. So we can kind of take the monomer and break the monomer down into parts too. So the nucleotide is a phosphate group, a pentose sugar, meaning a 5-carbon sugar. It is either deoxyribose or ribose. And then finally, a nitrogen-containing base or a nitrogenous base. It's that last one that tends to really confuse people. They get it mixed up with carbohydrates a lot, but the nitrogen should tell you that it is not a carbohydrate, not related to carbohydrates. It is a base. It will accept hydrogen ions. So again, the only DNA and RNA, the main difference, there's 2 or 3 main differences, but one of them is the sugar that they have in their nucleotide. So DNA has deoxyribose, RNA has ribose. The only difference is an H versus an OH there. Don't need to memorize the difference, but know that it is quite a simple difference. The nitrogenous bases themselves have 2 different families. I've never seen them ask questions on this, but they theoretically could. On pyrimidines and purines, I wouldn't really worry about it too much, but pyrimidines are bases with a single ring, so cytosine, thymine, and uracil. And then purines are the bases with 2 rings, adenine and guanine. You can see the mnemonic up there if you want to try to remember these, but you do not really need to. Adenine, guanine, and cytosine are in both DNA and RNA, but thymine is only in DNA and uracil is only in RNA. We get a bunch of these nucleotides and you string them together and you get a polynucleotide, or basically a nucleic acid. It's just covalent bonding again, this time between the sugar and the phosphate. It gets its own fancy name. This is where the phosphodiester bond comes in, if you remember from the Kahoot. But we're just linking them together specifically at this spot. But because we're linking a sugar to a phosphate instead of 2 of the same thing linking together, it leads to DNA having 2 distinct ends. On one end, it's going to end in a hydroxyl group. That's called the 3' end, because it ends on the 3rd carbon. And the other end is going to end in a phosphate, which is going to be the 5' end. So that gives it some interesting properties that we'll talk more about in DNA replication. It means that we can only grow a strand in a single direction. For now, I wouldn't worry too much about it, but knowing that, yes, the 2 ends are distinct is good. So we get all of these linked together, and we can fuse the 2 strands to actually make a DNA molecule. DNA is pretty much always double-stranded, but RNA is usually just a single strand. So that's one way you can figure out if it's DNA or RNA. If it's single-stranded, it's almost certainly RNA. If it's double-stranded, it's probably DNA. The other thing to really look for are the bases. If you see uracil, you know it's RNA. What holds the strands together is hydrogen bonding. So again, those hydrogen bonds coming in clutch. It is nitrogenous bases forming those polar bonds that then can hydrogen bond and hold the double helix together. This is a great example of one hydrogen bond being pretty weak, but if you get a bunch of hydrogen bonds together, it can actually hold its structure. RNA is weird. Sometimes it's single-stranded, sometimes it's double, sometimes it has a fancy shape that has a purpose, sometimes it doesn't. We're going to talk way more about it later. For now, no, it's single-stranded and a little weird. The way that we actually form those hydrogen bonds, it's not just any two bases can make a hydrogen bond, it is specific bases. So in DNA and RNA, C always pairs with G. So cytosine always pairs with guanine. And in, I don't know why that text on this is so weird. The difference comes in the other base pairing. So DNA has thymine and RNA has uracil. Whichever one they have will make a bond with adenine, and they just don't have the other nucleotide. So A pairs with T or A pairs with U. It will do both. Lastly, almost lastly, we get the anti-parallel structure. So because of the way that you have to orient the molecules for the hydrogen bonding to occur, you end up having the DNA strands being like flipped opposite, where if you look at the top of the DNA strand, on the left side it's a 5', on the right side it's a 3'. So on the left side it's a phosphate group, on the right side it ends in a sugar. That is going to be giving them some properties. Again, later you'll have to wait for genetics to really talk about this. For now, just know that DNA strands are anti-parallel. They run opposite. They end in opposite lengths. So, to sum up, RNA tends to be single-stranded, and it has cytosine, guanine, adenine, and uracil. DNA tends to be double-stranded. It has cytosine, guanine, adenine, and thymine. Instead, A pairs with either T or U, depending on what they have, and C pairs with G. Last thing I want to talk about, hopefully you know what ATP is, or at least heard of it before. It stands for adenosine triphosphate. What you may not know is that it is basically just a nucleotide. It's a nucleotide with an extra, or with two extra phosphates added on. It's adenine, so the A in DNA, or not the A in DNA, but the A that is part of DNA. Ribose, so the sugar from RNA, and then 3 phosphates. Usually, if you're making a nucleotide, you just have 1 phosphate. So if you add 2 more, you get ATP, which is a very high energy molecule. Essentially, this linking of the 3 phosphates together is kind of an abomination, and they want to split apart from each other as soon as they can. When they do split, they release energy. That energy can be used by our bodies for various tasks. We'll talk plenty about ATP in Unit 3. For now, I just want you to remember that, hey, ATP is actually basically just a nucleotide of RNA plus 2 phosphates. It is explaining, yeah, you can cut it, take off a phosphate, you get a bunch of energy that can do work. We're going to talk way more about this later. We'll go through those in class, and that is it. The last topic of this unit is basically proteins and enzymes, which will take us all the way to the test and may not have video lectures. We'll see. Wrong one. But look, Inception. That's all.